Good evening, and thank you for coming to the Los Alamos Council on Cancer's Fall Seminar. This year we're having Jessica Sims talk about breast cancer treatments. So our guest presenter tonight, Dr. Jessica Sims, is a local. She graduated from Los Alamos High School. She earned her bachelor's degree in biochemistry from the University of California in San Diego. And while completing her bachelor's degree, she worked at Vertex Pharmaceuticals in a high-throughput screening group, as well as in the laboratory of Dr. Robert Tukey, where she studied mechanisms of cellular defense against oxidative stress and toxicity. Sorry if I murdered that. Dr. Sims then attended Tufts University in Boston for grad school, where she worked under Dr. Dan Jay and studied novel mechanisms of breast cancer invasion and metastasis. After completing her PhD, she moved to Cedars-Sinai Medical Center, where she continued her training as a postdoctoral scientist in the laboratory of Dr. Lali Medina Kawe. While there, she worked on development, testing, and validation of a number of targeted breast cancer nanotherapies. After the completion of her postdoc work, she continued on in the Medina Kawe laboratory as a project scientist, where she is working on completing the preclinical work of several targeted nanotherapies, as well as studying new potential targets for the development of additional therapies. She's also served as a scientific consultant for EOS Biosciences, a startup company based on the technology that was developed in the Medina Kawe lab. Jessica is also active in the graduate program at Cedars-Sinai, where she's co-director of one of the graduate classes, as well as a mentor to the students in the lab. Welcome back to Los Alamos, and we look forward to hearing you.
What I really like to be talking today with breast cancer is about where we've been, where we are now, and where we're hoping to go. A huge part of continuing to develop more successful therapies is remembering our past and trying to better understand what has worked and what hasn't and why. So I'll be covering a number of these to help introduce some of the next generation of therapies that we're developing in the field right now. Really quickly, this is my required interest disclosure. I am paid consultant with Biosciences as well as a stockholder in the company. I will be briefly discussing some of the research of the company starting at the end of my talk. All right, so moving on to the more interesting things. Cancer is often thought of a very modern disease. It's something we see more due to some aspect of modern society, and it's really, cancer's always been with us. It's been, as long as we've been people, cancer's been present. Breast cancer, due to its location on the body, it's been much easier to detect without a lot of our modern imaging. And some of the earliest recorded cases actually date all the way back to ancient Egypt. In the Edwin Smith Papyrus, there's a note from an Egyptian physician that simply says once a breast cancer tumor has become more advanced, there's simply no treatment. There's nothing that can be done for these patients. And as we move forward to the ancient Greeks, Hippocrates, among many different things, did study cancer and was interested in these, and tried to understand why, what caused cancer, and his conclusion was it was due to an imbalance of the tumors in the body, the four different tumors that are important, that the balance of them kept you healthy. And this is likely due to the fact that once tumors were able to be outwardly observed by the physicians at this time, they were quite advanced, they'd be lactic, they would have a very necrotic appearance to them, leading him to think this was a cause from black bile. Now, Hippocrates is also credited with naming cancer. It's based off the Greek name for crab, or parthenos. There's probably two reasons he named this. First, from the observation of cancer slowly progressing through the body, like you might imagine a crab crawling along, as well as more advanced tumors often having protrusions similar to the arms or legs of the crab. Now, over the next few centuries, there were many theories in addition to the humoral imbalance as to what caused cancer. Some of them made some sense, some of them less. With everything from, you had some sort of physical injury, there was a virus that the person had, and it moved to there was some sort of clotted milk left in the duct, and simply there were even theories that you got cancer because you were scared of getting cancer, which is a bit ironic. And there were many different therapies that people tried. Most of them were just simply to relieve the pain and help the patient survive a little bit longer. These also ran the gamut from just religious prayers and rites to different balms and ointments that contain things such as mustard seeds and all the way to things that are a little bit more toxic than that. But none of these really cured the tumors. They would just help the patient for a short period of time. And for things especially like breast cancer, that it was obvious and not really apparent where the tumor was, you did have a few physicians occasionally trying to surgically remove these tumors. Now this time this is would be a little dicey. We don't have any of our modern techniques. None of these physicians knew about things being sterile. We didn't have ways of monitoring the people afterwards, and so you have a lot of morbidity and mortality from these patients in the attempt to remove these tumors. So this wasn't very popular. There's not any documented cases of this actually curing any of the cancer. But even so, as we move down to the 1750s, the theory that breast cancer actually came as a local disease and it wasn't a full systemic disease finally became much more widely accepted. And so the thought that, well, if we can find it while it's small, we can remove it from the breast, then we really reduce the chance of this patient surviving. And as with a lot of things, a little is good, so more must be better. So the surgeons throughout this period would initially take a small amount of the tumor, and they would take more of the tumor, and then they would take the surrounding breast tissue and the muscle tissue, and the whole thing kept getting bigger and bigger, and led to the culmination of this in what we call the radical mastectomy, which was really credited to the late 19th century surgeon William Halsted. And this quickly became the gold standard for breast cancer treatment. It was really the only thing they had was to try to remove the tumor to cure the individual. Now, while sometimes this did treat breast cancers, it also was a horrible procedure for these women to undergo. It left them scarred and often really reduced their quality of life. So a lot of people chose to not do these surgeries and to try to seek out other ways of treatment. And fortunately, at the end of the 1800s, radiation was discovered. And by 1903, amongst the other things that radiation could do, it was reported that it also could help treat cancer. Now, this was a rocky field for a while. A lot wasn't understood about radiation, particularly things like radiation poisoning. And a lot of different treatments were tried, and slowly over time, they found that things like small little doses of radiation was much better than one large dose of radiation. You got the same therapeutic effect without some of the unwanted side effects. And then finally, in the 1970s, really important clinical trial was performed, and they compared what we consider breast-conserving surgery, where only the tumor in a small area were removed, followed by radiation therapy against the radical mastectomy. And it was actually found that they both had very similar results, but the patients were much better off with a much smaller surgery. But radiation still was and is a very popular therapy option for breast cancer and other cancers. As we move into the 1990s, the whole procedure became much more sophisticated. We were able to treat it in a much more 3D format. We used multiple beams to help direct what we were interested in treating, so only the tumor received that really high dose of radiation we needed to kill those cells, whereas any surrounding tissue didn't receive that much radiation. We were reducing a lot of damage, as well as help with the overall patient morbidity. And the other big thing that entered the scene in the 1900s was after World War II, a number of research began to occur on some of the compounds that we use for biological warfare. And we found that In addition to the not so good aspects of it, a number of them had anti-tumorigenic effects. Now, a lot of these initial trials and experiments weren't done on breast cancer, but their success in those other tumor types quickly grabbed the attention of the other cancer fields, and these different therapies were tried in different orders and different combinations. And what was found with breast cancer is the most successful combination of chemotherapy is what we call CMF. This is still sometimes used. It's the combination of cyclophosphamide, methotrexate, 5-fluorouracil. It's quite a mouthful, which is why it's called CMF. These are 3 different drugs, but they all essentially accomplish the same 3 things. They affect the DNA of the cell. They prevent the cell from being able to divide by interacting in different ways with the DNA and end up essentially killing the tumor. And the success of these chemotherapies led to a lot of research in other drugs, other compounds. There were big searches through natural compounds as well as chemically synthesized compounds to try to find other things that were successful in treating these cancers and had even better cure rates than what we saw with the CMF. And some of these include the anthracyclines, which are doxorubicin or epirubicin, which also affect the ability for cells to divide by interacting with the DNA. We have taxanes, such as paclitaxel and docetaxel, which actually interfere with the structure of the cell, which keeps this, which is important for the division of cells, as well as some other things that keep the cell healthy. It's also that there are a number of antimicrobials that, along with killing your bacteria, are anti-tumorigenic. So, starting in the 1970s to the present, there have been literally hundreds of clinical trials looking at these different therapies, looking at different orders and different combinations. And because of this, the gold standard has shifted many, many times, and more it's changing based on what your tumor type is and your time of diagnosis. And I'll discuss some of the tumor type aspects in a few minutes. But unfortunately, overall, most of these gains were incremental. And as I have here, we have a few graphs showing the combinations, showing the relative mortality of these patients over time. And we have untreated patients versus patients treated with CMF, that triple therapy I mentioned first. We were able to decrease mortality by about 4.3%. It's not huge, but that's important for that 4.3%. And then comparing that treatment to anthracycline, we have a further gain, as well as for another chemotherapy, we now gain another 5.1%. So we're slowly inching our way down. But ideally, we want it to be a huge difference. We want to be seeing 40, 50, 60% reduction in mortality. And so that the cancer field's really working to try to improve this. And what we found at this point is that one of the best things you can do is early detection. The earlier that the tumors are found, the more successful these treatments are. But what we want to do is work towards finding therapies that it doesn't matter when we diagnose you. We can find something that will work for you. So the cancer field in general, we really tried to understand the different tumors, understand what causes tumors, and this is a very famous figure in the cancer field. It's from a paper that came from Bob Weinberg's lab. He's a cancer researcher out of Boston, and it's called the Hallmarks of Cancer. And what's important is you can see here, this is demonstrated as a circle. And it's because there's no one starting place for tumors. It depends on what the tumor type is, and all of these are important for its growth and throughout the entire cycle of the tumors. So I'm going to briefly talk about each of these aspects and some of the importance that they have in developing therapies and what we've used and what's worked and what hasn't. In a normally functioning tissue, the cells all talk to each other. The tissue wants to be as healthy as it can to keep the organism as a whole healthy. And the cells will tell each other, you need to grow, you need to stop growing, you might need to undergo some sort of programmed cell death just for the betterment of the entire tissue of the organism. And the cells have all adapted to finding this, listening to these signals and to talking to each other. And cancer cells are able to find a way to simply ignore this. They don't listen to what the other cells or the other tissues are telling them, and they continue to grow, so they'll ignore signals that tell them to stop growing, similar to what we have here. They also do need signals to tell them to grow, so they've learned to make their own signals. Or we found more and more that they're actually able to co-opt their neighbors. That actually, you're wrong, I should be growing, you should make these growth signals. And also, they'll get signals telling them to undergo programmed cell death, which is something we call apoptosis, from other surrounding cells and surrounding tissues, as well as from themselves. Our cells are quite smart. We know if something starts to go wrong, they realize, you know what, it's better off in the tissue, I should just die. And so they undergo this programmed cell death. And even though there's things that have gone quite awry in cancer cells, they no longer have that internal message that for the better of the tissue, they need to undergo this apoptosis. So they're able to evade this process of apoptosis. The other really important thing that's down at the bottom is this limitless ability to continue to divide. Some of you may have heard of things called stem cells in the news, or research or other things that you've done. These cells are able to keep dividing forever and ever and ever. But once a stem cell doesn't make all differentiate, become something more specific, say like a liver cell or a lung cell, or something that has very set job in the body, it only can divide a few times. And after that, it becomes what we call quiescent, or just and stops growing. Now, these cancer cells have to be able to also ignore this signal. They're able just to keep dividing, even though normally they shouldn't be able to do this. So we have this tumor, different size, that keeps growing, doesn't want to die, but even though it can make its own growth signals, it still needs nutrients to survive. So it needs to be getting this from the organism, from the blood system, and it has to be able to encourage the growth of blood vessels into the tumor area in order to survive. So a really important part also is seeing angiogenesis. Angiogenesis is just the growth of blood vessels to the tumors. And this was actually a very exciting and then disappointing area of the cancer field. Another very famous researcher, Judith Folkman, really pioneered the field of angiogenesis. He was the first one to point out the importance of blood vessels to cancer growth. He was actually pretty much laughed off the stage initially. Most people thought he was crazy, but he actually proved that this is a very important part. And once that was accepted by the field, the cancer field became very excited that we found the answer. If we could just cut off the blood supply, cut off the root to the cancer, we'd have a cure. And so there was a race to find a number of different therapies that would stop this growth of the blood vessels, would starve the cancer cells of what they needed. But unfortunately, they did not pan out like we wanted to. And I think it comes back to this idea that you can't just knock out one piece of this. You have to get the entire cycle. And lastly, the other aspect of tumors, which is one thing that we really need to address because it causes the majority of the deaths in cancer, is tissue invasion and metastasis. As tumors grow, they run out of space, they run out of food, they want to move on to new areas, and they're able to essentially invade through the tissue into the blood vessels and other vasculature of the organism and travel throughout the body until they find a nice comfy home where they're able to establish new tumors. And this was often thought to be a problem in more advanced cancers. Initially, the tumor was happy in its little home, and it didn't need other areas to live until it got too big. We're actually finding that it's not the case more and more, that even small tumors are seeking out new areas to colonize in the body, and that addressing the issue of metastasis is important for pretty much all stages and throughout all tumors. So that's just a quick introduction of a lot of the areas of cancer, what we're trying to address. We're trying to have therapies that fit all of these areas. But homing more into breast cancer now, even though it's often looked at as a single disease, it's actually vastly, vastly different. You will have breast tumors that are more similar to the liver cancer than they are to another breast tumor. And the breast cancer research field has been trying to further stratify tumors into different groups so that we better understand them and so that we can develop better therapies for them. And so one of the first things that was identified in breast cancer is there's a number of them that are very dependent on hormones, such as estrogen, which would make sense. It's in tissue which hormones are very important for. And this led to the development of a few anti-estrogen therapies, which I'll talk about later. Another thing that was found to be upregulated in another subgroup was a receptor called HER2. And that also led to a race to find therapies for these HER2 tumors, and I'll also discuss some of those in a few minutes. And the third group is what we often call triple negative. They're not estrogen receptor positive, they're not progesterone receptor positive, they're not HER2 positive. Well, usually it's not good to define something by what it's not. And so the field's really been working to better understand the triple negatives and not just simply call them what they're not. And this has now been further stratified quite a few times. What I have up here is vastly simplified from what's in the field now. These often can get divided into 6 or 10 different groups. But one of the primary divisions is whether or not this EGFR or one receptor is positive or negative. And so these different groups have really helped us better understand how we can treat each of these patients. So often, for anyone who may have had breast cancer or knows someone who's had breast cancer, you've probably been screened for these things as to whether or not you're one of these tumor types, and that's really affected what therapies you then have received. So another reason why these subtypes are so important is that they really do affect the outcome of the treatments and the survival probability of these patients. Now this is an indication of what the survival is of these people who are not treated. And what's interesting is our top line up here, so these are our estrogen receptor positive tumors. They actually have the best prognosis, and then followed by our triple negative down here, and then our HER2 positive tumors have definitely a very poor prognosis, and then this particular form of the estrogen receptor positive, but they're also HER2 positive, have the lowest prognosis. Now this has changed a lot because depending on which tumor type, we have more and less treatments for it, but understanding this has really helped us realize where we need to be focusing a lot of our research. So this really led to what I consider to be the emergence of targeted therapies. Now often estrogen and HER2s aren't considered targeted therapies, but that's really what they are. When you have some of the general chemotherapies, like doxorubicin, those things are given systemically. They attack any sort of quickly dividing cell, which happens to be your cancer cells, but it also can be hair cells and gut lines. And a lot of side effects there because you're targeting a lot more than just that tumor. So in these targeted therapies, breast cancer has been a really great field for because of our ability to stratify it into these 3 groups and find treatments that are very specific for that tumor type. So for our estrogen receptor positive tumors, we have a number of different anti-estrogen and progesterone therapies for our HER2 and HER2 therapies, and more recently for our EGFR positive subgroup of triple negative, we have therapies for that as well. So going into a little bit more detail, targeting estrogen. Now, this group of patients is then further stratified based on whether or not the woman's undergone menopause. For premenopause women, we have both estrogen receptor modulators as well as downregulators. So for your modulator, you're essentially preventing the interaction between estrogen and the estrogen receptor. And for those of you who may not entirely know what I mean by a receptor, it's almost like an antenna. It's on the outside of your cell and receives signals from other places. Often there's some sort of a growth signal. And estrogen works that way. It's something that's coming from other parts of the body. It binds to that receptor and helps the cell grow and undergo whatever other processes it needs. And so these particular drugs just prevent this interaction. Then there's also the downregulators that they essentially pull in the receptor. They remove that antenna from the cells so they're no longer able to interact with any estrogen that might be circulating. Now these are both important for the premenopause because these women have lots of estrogen just normally cycling in their bodies. Now after menopause, we have what we call aromatase inhibitors, and they actually inhibit the conversion of another hormone, androgen, into estrogen, because there isn't much of that free estrogen around in postmenopausal women. And there's several drugs that are actually able to do that as well. So this subgroup, estrogen receptor positive, we actually have some great therapies for, and it's really improved the prognosis for these particular patients. Moving on to HER2. So HER2 is another cell surface receptor. It's another antenna. It looks out for regulators that may be coming from different parts of the tissue and the cells and the body. And it's found that HER2 is often upregulated on the cell surface of these cancer cells. And prior to these therapies, this group had an extremely poor prognosis. So this is just a little example. In a normal, healthy breast cell, you'll have a couple of these receptors. They'll get their HER2, they'll grow. But in the breast cancer, you'll have a huge number of these receptors on the cell. So they're pulling in as much of these signals as they can, causing lots of growth and inhibition of some of the apoptotic factors they'd be getting from other areas. So this field, because of the poor prognosis of the patients, people were desperate to try to find something that would work to inhibit this HER2 receptor. And the first drug that entered into clinical trials and then was FDA approved is called trastuzumab. It's also, its other name is Herceptin, and it was approved in 1998. This was actually over a 10-year process of development from when it was first discovered to when it was able to get FDA approved. Now this is actually based on an antibody. This was a rather novel idea at the time. And antibodies are what your body used to defend it against foreign invaders, viruses, bacteria, etc. But it's very good at binding to whatever bacteria it is that you want. So we realized that we could co-opt to design our own antibodies that instead of trying to detect a particular virus, it actually would detect that HER2 receptor. And there was a long process in the development of one of these that worked successfully, but once it did, it was quite revolutionary for the field. So here's a little diagram of an antibody. They're often depicted as kind of an upside down Y. We'll see a few more of those later on. And this is a picture of our RB2, and this is the part that's outside of the cell. This is the antenna that's looking for its signal. And once this antibody binds, it prevents it from receiving its signal. So it stops that, the growth signal, and actually causes the cells to die. And the success of this particular antibody led to the development of a later one in 2012 called pertuzumab that just binds to a different area of the HER2 receptor. So here's a little, this is purple here, but this one binds in a different place and actually prevents it from interacting with other proteins that are important for that cell signaling. And then there were some small molecules developed. Lapatinib is another one. That here, this is a small enough drug it's able to get inside the cell and interacts with the intracellular part that prevents the signaling there. Now, lapatinib is also used for other things. It's able to inhibit EGFR, which is important in triple negative tumors. And a very new idea to the field that also targets HER2, and this was recently approved, is something called TEM1. And as you can see, it also has the word trastuzumab in it. And so what we've started doing is combining our targeting antibody with some sort of a chemotherapeutic drug. So once this is given to the body, you don't have a chemotherapy that's traveling everywhere, but you've actually specifically targeted to your area of interest, and only that tissue receives this toxic chemotherapy molecule. And so it has that same binding function that trastuzumab alone does, but it then has this cytotoxic drug that further increases its efficacy. But even though we have lots of options right now for therapies, we unfortunately still have a lot of limitations. With general chemotherapy itself, we have a lot of off-target morbidity. There's a lot of toxicity, and a lot of patients very quickly develop resistance to that particular chemotherapy, requiring other therapies, additional therapies for that individual. And surgery is very effective for some people, but not for everyone, especially because it's impossible to know if you've got an entire tumor or metastases have occurred or other areas. So that's why you'll often see surgery combined with chemotherapies or with radiation to try to make sure you get anything that the surgeon wasn't able to do. And that really, in a lot of areas, it's very much of a standard of care. A lot of the patients that I work with have undergone some of these chemotherapies first prior to surgery or vice versa. And like I mentioned, radiation is often combined with surgery. It's very rare that you only see radiation treatment with breast cancer. But you do still see local and distant recurrences that can happen after radiation therapy, as well as just some of the side effects of patient morbidity that is associated with that. And one other issue that happens with radiation, not as much with breast cancer, but you do have development of secondary tumors. You actually see this much more in children that have been treated with radiation, but it predisposes them later in life to develop other tumors. But as our technologies improve with radiation, we're getting much less of the collateral damage. We're mainly just exposing the tumor area to the radiation, so this is happening less and less. And last, we have our targeted therapies, the anti-HER2 or anti-estrogen receptor. They also will have some off-target effects. While there's a lot less tissue that have these receptors, there still are tissues that have HER2 upregulated, or unfortunately one of them is cardiac tissue. And same with the estrogen receptors. And what we're finding as these therapies have been used more in the clinic, as you've seen before, a lot of them are very recently approved in 2012, so we haven't actually had a long time to study these yet. We're seeing patients develop a lot of resistance to these therapies. So we're trying to better understand what we need to do in addition to some of these targeted therapies to keep the patients in remission. I wanted to go into a little bit more detail in the field of targeted therapies. This is my area of interest. This is where I do a lot of my research. In theory, the idea of targeted therapy sounds great. It seems like something we can easily be able to do, but there's actually a lot of challenges. But the main goal of targeted therapy is to develop some sort of a compound that only binds to your tumor cell. It doesn't have any effect on the surrounding normal tissue or any of the blood vessels or any other parts that you're only targeting your tumor tissue. So there are a number of different challenges that we have to keep in mind with this in developing these therapies. And first, are much more related to the overall body as a whole. We have to be able to very specifically target to the tumor. When it's delivered systemically, most of these therapies are still the point where they have to be injected, so they go through your entire circulation system. They have to be very stable. Your blood and your circulation is spent millions of years going through evolution to get rid of foreign objects. They don't want these unknown things there. They get excreted very quickly. You mount an immune response against it. So we have to do everything we can to make these particular compounds be able to last in the blood and to be able to remain in circulation long enough that they can reach their target area. If you inject someone with this and their kidneys promptly cleared, it's not going to do them good. And as well as, like I mentioned, you want to have as little immune response as possible, because there are some therapies out there for other cancers, such as melanoma, that can cause really, really big immune responses. So we've been working with ways to essentially hide our therapies, make them invisible to the immune system. So even if you manage to accomplish all of that, you reach your tumor site, now you have a whole other set of challenges to overcome. You have your drug of choice here sitting outside of your cell. You need to get into the cell. Cells don't just take up anything. There's a number of different mechanisms for things to get in. Anything that's really, really small that can get into a cell easily, that's not going to survive in the larger body. It's going to get cleared by the kidneys before it can get anywhere. And so the primary way that cells take up these larger compounds is a mechanism called endocytosis, or essentially cellular eating. They take up these compounds in these nice round vesicles that almost function as the stomach for the cells. And once these things have gotten into the cell, they become more and more acidic, so they can digest things just like your stomach does. And this is good for the cell if it happens to take up something that's bad, that is not healthy for the cell, it destroys this object. As well as if it is some sort of cellular food, it breaks down those proteins or other things that the cell can then make and build whatever it needs to out of it. So if your drug is stuck in there and acidifies and gets destroyed, it doesn't do you any good either. So you need to find a way to get out of this endosome, this cellular stomach, before your drug is destroyed. And then finally, you have to be able to then deliver this cytotoxic agent to its area of interest, which is often the nucleus and has to do with cellular division. And then having this drug be really successful in resulting in cancer cell death. So the two ways that the field have primarily tried to do this are these two different therapies. The first are these antibody-drug conjugates, and the second are through nanoparticles. I'll talk about both of those for a few minutes. Now, this antibody-drug conjugate may sound slightly familiar. This is essentially what one of the therapies I brought up earlier called TDM1 is. And this is a very, very basic schematic of it where you have, once again, this is your antibody, your Y-shaped protein here, and it's got some sort of a chemical linker or connection to whatever your drug is. And so the idea is your body bodies very used to antibodies. It's not usually treated as a foreign substance. It doesn't get cleared very quickly, and antibodies are incredibly good at targeting to whatever they've been designed for. And we've gotten very good at designing antibodies to target pretty much anything you can think of. And then the other side of these are nanoparticles. These are a little bit more of what I would consider a designer drug. They come in many shapes and sizes, and you can do lots of different things with them and give them different characteristics you need for your tumor killing. And I'll discuss those in more detail later as well. So first I want to talk about the ADCs, or the antibody drug conjugates. We do have one that is approved, that TBM1, so that is in the clinic now, and it targets HER2 and delivers the entansine. There are several more that are undergoing clinical trials, and there are a number of pharmaceutical companies that are trying to develop other of these agents. This is actually a huge area of development. And they're all based off of this general idea where you have your antibody here, with the drugs linked to it through these little linkers. Now this is a much more accurate picture of what I had before. Usually it's not a one-to-one ratio. You don't have one drug molecule for one antibody. You'll actually have quite a few for each of these antibodies. And then moving on to the targeted nanotherapies. First question we often get is, what is a nanoparticle? What's considered a nanoparticle? And really it's anything that's in this size range here of 1 to 100 nanometers. And there are a number of different compounds that have been designed, not all of them are for medicine, but they include things such as micelles and liposomes, which have some sort of outer shell that is hydrophobic. It keeps the water away from it. It's able to keep whatever you have inside nice and safe. There are dendrimers, which are rather similar to these liposomes, but they're much more they have a much more different shape in that they don't have that coating. But what people will do is you'll functionalize the outside of all of these tentacles with whatever drug you need and with whatever properties you need for your nanoparticle. And there's also things like the gold nanoshell, the quantum dot. These are things you'll find more in chemistry. They're very good for imaging. We don't see this much in therapies right now. And then finally, there are polymers. Polymers really function as a scaffold or a structure for designing whatever drug you might be interested in. And like I mentioned, these really are kind of considered a designer therapy. You go find your favorite chemist and consult them on how you can combine some of these elements to get the drug that you need. And we try to incorporate many different things, including having it be biocompatible or biodegradable. Once we've delivered our drug of interest, we don't want the rest of the nanoparticle to cause morbidity or other side effects. And the size of the nanoparticle, that 1 to 10 nanometers, actually is the sweet spot that we found. It's large enough that it stays in the circulation. It isn't cleared quickly by the kidneys, but it's small enough that you're able to be quickly taken up by the cancer cells. And in these therapies, we're also trying to incorporate targeted delivery as well as immune system evasion. We've gotten very good at essentially being able to disguise our molecules by putting special proteins, special little signals on them that the immune system doesn't see. And due to the many characteristics of these nanoparticles, it allows us to deliver a large number of different payloads. So there are a number of these also in the clinic or other clinical trials. A lot of them are this more micelle-based idea. One of the most well-known is Doxil. This is targeted doxorubicin, which is a pretty well-known and highly used chemotherapy. There's also Abraxane, which targets paclitaxel, myosin, so you can kind of head down the list. And one interesting thing to point out here is that most of these drugs are all well-known chemotherapies that have already been approved just for systemic treatment anyway. And so often the thought is, well, why are we bothering to put them with these nanoparticles if we can already use them as just a regular chemotherapy? And the hope is that by specifically targeting them, these chemotherapies are no longer going to the stomach and hair and other quickly dividing cells, but we're just seeing that toxicity in the tumor tissue. And also, because these chemotherapies have already been approved by the FDA, it's actually much faster to move them through the process. That's been a very frustrating thing for both patients, for physicians, as well as for researchers, is how long it takes for a drug that appears to be very successful to finally get into clinical trials and then into the clinic. And so if you have something that's already been, part of it's already been clinically approved, you can move through that process much quicker because you only have a change in your delivery. So in developing a new and novel nanoparticle, here are some of the challenges I mentioned before, but we need this to be efficient. We need to be able to stay in circulation for as long as possible. We need it to evade the immune system and not target the other tissues. And we need to, once it gets to the cells, we need to be able to get the nanoparticle into the cell, and then to have it release the drug, and to also to keep the cell in the drug, keep the drug inside the cell. This is a challenge that a lot of people are finding some of these nanoparticles. Cells don't like toxic elements in them. They also have found over evolution have found ways to quickly kick these things out. There are these drug efflux pathways, and so we need to find ways to make sure whatever drug we put in isn't properly kicked out by the cell before it's able to have this toxic effect. The group that I work with took a rather different approach to this. Most of what I've talked about have either been antibody-based or some sort of a chemically based idea. And we wanted to develop a particle that's much more protein-based, that's much more familiar to the body, familiar to the cell, and enabling us to have easier ways of producing it. So our initial interest was with targeting HER2-positive breast cancers due to the need in the field. But as we really looked into the HER2 field and what's targeting and what wasn't working, we realized that the HER2 doesn't function alone. It needs another antenna with it. And so this other partner is called HER3. And HER3 gets a different signal, what we call the ligand. And so we were interested in trying to target that side. And so what we did was developed a therapy that we essentially use that signaling molecule, and that's our way of targeting to these cells. And one unique thing about these receptors that a lot of people haven't taken into account is that there is just a general process that the cells use to stay healthy, where as soon as there's been a signal acquired by these receptors, by these antennas, they actually get internalized into the cell. The cell takes them in, reprocesses them, and then decides whether it wants these signals back out again. And that this is a very normal process of endocytosis, and it's a really easy way to get your drug in, where you're not having to use some other pathway that can cause other problems. And so by using a signal that the cells are used to, that as soon as you have that signal hit your receptor, it causes the receptor to go inside, we're using this kind of as a hack, as a way to convince the cell to take up the drug without realizing what it's doing. So and so now we have essentially our targeting part. So we can get inside the cell. Now we face with the next problem of you end up essentially in that cellular stomach. You're going to get dissolved. You need to get out of that acidic area as quickly as possible. And so we turn to nature. There's no reason to reinvent something that somebody already did. And this is something that viruses are incredibly good at. Viruses get into cells in a very similar way. They bind to these receptors, trick the cell into taking them up, but they also have to get out of this endosome or cellular stomach. And so we spent a lot of time looking at this Ad5 adenovirus. It's just one of the viruses that are often seen. And we found that there's actually one tiny little piece of this virus, which is called the penton base. That is what allows the virus out of this endosome. The rest of it isn't necessary. And so we decided just to follow that one tiny little piece and add that to our targeting molecule to allow our drug to get out of the cells. And as soon as you use any portion of the virus, people become worried because it does cause immune response and various things. But we're using such a tiny, tiny part of it, there's no issue with the immune system. And we've actually spent quite a lot of time testing that and verifying that. And so when some sort of animal or cell has been exposed as part of the penton base, it doesn't have the normal immune response you have if you were to see this entire virus. we then combine these two pieces to develop our targeting portion of our nanoparticle. So we have our three parts. We have that binds to the receptor, that targets to your cells. So it's our, it's essentially like our address, it gets us to where we want to go. And then once we're in there, we have this penton base that allows our nanoparticle to escape and to then be able to deliver the drug to whatever part of the cell we're interested in. So this is great, but right now we don't have anything toxic. This isn't useful to us yet. So we've added a large tail. This is positively charged. This just gets into the chemistry side of things. It allows us to bind up a lot of different drugs. And the benefit with this particular compound is it's really just a long string of proteins. It's something that can be made in bacteria. It's something that can be produced in very large quantities, which is the issue with a lot of the nanoparticles, is you'll have some very complex structure that's chemically made that is very, very expensive to make. And so it becomes cost prohibitive to actually scale this up to the point where we can treat anybody. It's great for your research, but it doesn't really help anyone in the clinic. So now that we have this targeting portion, we need to decide what it is we want to do to add some sort of a toxic payload. And how we designed this first targeting portion, it will spontaneously self-assemble into this nice round particle with whatever drug of interest we have. In our case, we've been very interested in targeting doxorubicin. And so we use this particular drug, we call it our eosome, and we tested this both in cell lines and in small animals and had very good success in either killing tumor cells or reducing tumor size. We've shown that it's extremely stable under physiological conditions, pH, salt, temperature, and those different components, as well as targeting very successfully to the HER3 portion of the tumors. So then moving on, we have a lot of different therapies. I talked about a lot of different things, including surgery, radiation, hormonal therapy, chemotherapy, and then more specifically targeted therapy, which is often the targeting of chemotherapies. And what we found is that really successful treatment of breast cancer involves understanding the breast cancer type and where the patient is, and deciding which of these therapies is going to best help that particular patient. And more that we need to use multiple of these therapies to prevent recurrence and to prevent resistance from happening. And also, by carefully choosing these and continue to improve things, and as more and more not using the systemic chemotherapies, but moving into much more targeted chemotherapies to really reduce the side effects of patient morbidity, so that going through the process of chemotherapy is no longer a horrific experience. It really is for a lot of people. And as we continue to better understand breast cancer, understand how it functions, why it grows, why it develops resistance, we'll be able to develop more and additional effective treatments for treating breast cancer as well as other cancers. And just some quick acknowledgements. This is I work under as Amy mentioned. This is the lab members. This is EOS Biosciences, the small biotech that we have trying to bring some of this research to clinical trials, as well as some of my collaborators that I work with.
So if you have questions for Dr. Sims, you can write them on the card, and we'll ask them as we get this done.
So I have a couple of questions here, they're all very good. There's two of them that are very similar, so I'll try to address those at the same time. And they have to do with screening and mammograms, which is actually been something that's been in the news a lot. It is an area of great discussion as to how much and what we need to do. And while I'm not a clinical oncologist, please don't, I know what the field's been doing and what's often recommended. A lot of these are going to be based on your family history. If you have cancer or breast cancer in your family, you want to stay with the really rigorous screening, once a year or whatever. You want to have a relationship with either your primary care who knows a lot of this or your oncologist to be very diligent in watching this. For younger women, I think the screenings, the mammograms, every 2 to 4 years is much more normal. They've really increased the amount of time. The older you get, it's really recommended more and more to get more often, or to get mammograms more often. But also a lot of these you can tell yourself. The self-exams that I'm sure all of your physicians will tell you about doing. This really is a good way just to keep an eye. You know yourself better than anybody else does, and to notice any changes. The things happen, even if it's something small, then that's a good thing to go and just have someone check it out and make sure everything's fine. The insurance world's going to make that more difficult depending on what they cover, but that's where I think it's very important to know your family history, have a relationship with a physician. That if they really do feel it's important for you to be having these mammograms more often, they usually will help whatever needs to get covered by the insurance get covered. So the breast cancer, I have another question here about breast cancer in men, and that actually I do not know a lot about. It's actually my grandfather-in-law did pass away from breast cancer, so it is something that's out there. It's not talked about a lot. From what I know, most of these are probably what's considered triple-negative tumors. They're ones that have some of the other receptors upregulated or different mechanisms that we're still really trying to understand. But that actually really is a very underserved area of the breast cancer field. And then my last question, I'm doing my best with the handwriting. Oh, okay. So, I guess it's interesting that there's some of the breast exams and mammographies, and like I said, we still really do recommend, especially keeping an eye on yourself, and as soon as you have concern, going and seeing physicians, because catching things early really is your best chance for getting things cured quickly and for not having to undergo more treatment than you need. So then the second part of the question has to do with chemotherapy. And it looks like also has to maybe do some with apoptosis, whether or not chemo causes apoptosis in cells, which yes, it definitely does. But that still can happen in normal cells. The cancer cells are much more sensitive to these chemotherapies, and you'll have more cell death there. But really, a lot of the systemic chemotherapies, they work on quickly dividing cells. Most of them are either interact with the DNA, and the ability of the DNA, every time a cell replicates, that DNA has to copy itself. And there's a lot of cellular mechanisms that if that copy system is not working correctly, the cell will just kill itself, will undergo cell death. And occasionally the structure of the cells, the scaffolding, things that keep everything together are also very important in that cell division process. So almost all chemotherapies affect one of those two mechanisms, whether it's the structure or the DNA. And so that happens with any quickly dividing cell. So you'll see that apoptosis in the cell death in both areas. Now the caveat to that, which is why there's some issues in the resistance that you have with chemotherapies, especially ones that are affecting your DNA, is as these cells are replicating, things start to go wrong. You get mutations, you get strange things happening with the DNA, you have pieces that are associated with parts that shouldn't be, and any normal cell will go, okay, there's a problem, we're undergoing cell death, and that's fine. Well, these cancer cells have already really downplayed those cell death signals, and so even though something has gone drastically wrong with the copying of the DNA, they will still survive. And unfortunately, we have the wonderful world of natural selection that you'll have a whole host of these cells that try to divide, stuff didn't work out, they're still alive, you'll have that one cell that managed to get a mutation that is now given it an advantage, and it no longer cares about that particular chemotherapy that can survive. It's found a way around it. And then that will then be what later comes on to be that resistant tumor to that particular chemotherapy. And then we have to go into something new that this particular cell type can't get around. I'm going to stop on that too. Off target. Okay, this is actually a really good question as well. This is asking whether or not some of these therapies target stem cells as well as their daughter cells or their progeny. This can end up in, we're just mainly stick with the cancer side of things, otherwise it starts to get very complicated. There's been a lot of work in the cancer field with stem cells. What's the role of these stem cells? You're going to see it in any tissue that you have, some breast tissue as well. That they're really responsible for a lot of the tissue being healthy, and that they will eventually divide. These stem cells are quite amazing. They can divide and make two more themselves. They can become two stem cells. They can become one stem cell and a daughter cell. And even in some cases, they'll then terminally differentiate into two daughter cells. And these particular cells usually then don't divide anymore. And a lot of the therapies, what we are finding is that the chemotherapies are very good at killing these daughter cells, these terminally differentiated cells, but they're not very good at killing the stem cells. And so there's another working theory that a lot of researchers are trying to better understand is that some of your resistance and recurrence is actually due to the few stem cells that have survived. That you kill off all of the surrounding cells, all of the daughter cells, but you have your 3 or 4 stem cells that have survived, but over time they slowly were able to start replicating. And you end up with almost log phase growth where it's very slow growth and then it really shoots up as soon as you're able to get a lot of cells there, and that may be causing a lot of the recurrence. And so there are a number of people looking for treatments that really do target those stem cells. But there really isn't anything that's gotten into clinical trials yet there. Last part of the question, I don't think. So whoever this is, if you can ask me, I'm sorry, I'm not quite sure what the last part of that says. Okay, so these are 2 questions. The first one, do patients typically have just one subtype of tumor or several? This is another really excellent question. In theory, or initially, tumors were really thought to be a very clonal population. Like you have this one tumor, one cell that has gone totally awry, and it's built up this huge tumor around it. It's just essentially kept copying itself over and over again. But like I mentioned, this as the cells copy themselves, they're doing very quickly. The whole DNA replication process doesn't go through all the checkpoints and all of the normal processes that cells would, and so you start to get lots of these errors, lots of mutations, and lots of things change, and so you'll end up with these 2 cells that instead of being perfect copies, you have one that has this one characteristic and this other that has a different characteristic, and those then go on to divide, and so you end up with these subpopulations of this tumor cell A and this tumor cell B, and then B will then eventually go on and it may make a C and a D, and so you get lots of these different subpopulations, which is another reason why you'll see multiple therapies being more successful in a tumor than just one chemotherapy, because you're trying to find ways to make sure you're targeting these different subtypes. And so an antibody. So the antibodies don't do the killing. The antibodies are really a big signal for your body that there's something wrong. It's part of your adaptive immune system. You actually have 2 different parts of your immune system, your innate and your adaptive. And what it does is if it finds something, say the cold virus you've already seen, or something that you've been vaccinated against, which is polio, these little antibodies will bind to the bacteria, will bind to the virus or whatever it is, and it then signals to specialized cells that your immune system has made and and those actually go in and do the killing of the bacteria or get rid of the virus. And so the antibodies that are in the clinic, particularly the trastuzumab one, the anti-HER2 one, that it just binds to the receptor, it binds to that antenna, and manages to cause cell death. To be honest, the researchers got really lucky in this, it had this effect. And just by where that antibody happened to bind, you'll have this large protein where antibodies only bind to a little tiny portion of it, or where it happened to bind, it shut down the signaling. And the cells are so dependent on this downstream signaling from this receptor, these signals telling it to keep growing, telling it to keep dividing, telling it to ignore all these signals from everyone else to die, that it then causes these cells to finally undergo apoptosis and to no longer divide. It's also, we're also finding, as we're trying to understand some of this therapies, it's actually approved before we totally understood it. Because it is an antibody, the immune system, the circulation does recognize it, and it will recruit those specialized immune cells to that cancer cell. And those immune cells we're finding will also help to get rid of that particular cancer cell. And that leads to another area that's of interest. There's a lot of research in a lot of pharmaceutical companies where we're trying to co-opt the immune system to get rid of the cancer cells. Cancer cells, the majority of the time, have found ways to make themselves invisible to the immune system as well. Very often, when there is diseased tissue, you will have the immune system coming in to help clear it. But the tumor cells, even though they're really in diseased tissue, diseased area, the immune system ignores them. And so we're trying to find ways to wake up the immune system and tell them that this is actually something to get rid of. And so I think a little bit of those targeting antibodies do start to tap into that. There's a lot of other more sophisticated therapies that are seeming to be very successful in getting the immune system to help out. But they're not perfect, and these cells, unfortunately, are very smart, and they're able to find ways to start to ignore those antibodies, and so the bodies, we can use that antibody to also bring in drugs, which is why we have more and more of those antibody drug conjugates starting to hit the market. Okay, I think that's it. Anyone else?
Thank you very much, Dr.